Hi, everybody. Thanks a lot. Thank you for a generous introduction. It's an honor to be here, particularly because this is the Taylor White Seminar, which honors two scientists who have influenced my work greatly. I'm sure you don't all need to be reminded about the famous 1990 White et al. Paper closing in on, I don't know, 1 billion citations or something now. And, so I actually went on Google Scholar. I knew I'd cited that paper, and I went on Google Scholar and tried to figure out how many times I'd cited it. And I have personally contributed far less than half of 1% of but half of 1% of 1 billion is a lot. Anyway, thank you for having me. I'm honored to be here. So today I'm going to give you a talk about that mixes my recent interests in fungal comparative genomics and more long-standing interests in phylogenetics and the evolution of wood decay in agaricomycetes. I'm going to do the acknowledgements at the beginning because I may run out of time. I've had a lot of help from JGI up the street, so Igor et al. Dimi Flutis and Laszlo Nagy used to be in my lab, but they've moved on to bigger and better things. Many collaborators, and the NSF has funded the lab portion of my work, so thanks to them. Okay, so the fungi I work on mostly are the agaricomycetes. You all know them because they're the mushroom-forming fungi. They're really beautiful. One of my interests has to do with evolution of morphological diversity. But what I'm going to talk about today is the evolution of ecological diversity, because these things do really different important things out in nature. There are some biotrophs, including, this is the major concentration of ectomycorrhizal fungi. But there's also saprotrophs, decayers, and that's what we'll focus on today, particularly the wood decay fungi. So this is really the group that includes the premier decayers of massive woody substrates, at least in temperate ecosystems. But lots of different ecological modes. And sorting out the evolution of these modes is really hard because it's just tremendously messy. So one thing you can take away from this slide right away is that these fungi toggle between really different lifestyles fairly easily because adaptation in fungi is biochemical in nature, right? So they switch between being mutualists and saprotrophs fairly fluidly. But today we're going to focus on wood decay, the wood decaying saprotrophs. And wood, as I'm sure you all know here, it's a pretty complicated material, right? So it's this mixture of carbohydrates, cellulose, and hemicelluloses, which provide nutrition for fungi, the carbon and energy source. And all of this is embedded in this matrix of lignin, this very complicated heterogeneous polymer that we think is not an energy source for fungi, but has to be overcome or circumvented to get at the carbohydrates. This is also a major barrier, of course, to cellulosic ethanol production. So it's a complex mixture of really different components. Cellulose is really old. So this shows the origin of cellulose synthase genes in plants and dictee from various bacteria. This shows the occurrence of cellulose synthase families across the archaeplastids, the group that contains red algae, green algae, and plants, and then also transitioned into some other groups of eukaryotes. So cellulose, old. Lignin, also old, but less old. So we think maybe around 400 million years old, something like that, so in the land plant lineage. Now there's also some suggestion that there might be some lignin synthesis in some red algae, but it's fairly modest component and clearly of convergent origin. So this is really a relatively recent innovation limited to the tracheophytes as far as we know. So wood is complicated, and fungi have a lot of different enzymes involved in decay of wood, so lignin degrading oxidoreductases. We'll focus on the class 2 peroxidases today. Hydrolytic enzymes that attack carbohydrates. LPMOs are lytic polysaccharide monooxygenases that we'll talk about as well. Bunch of accessory enzymes. It's very complicated, and this is not my comfort zone. So biochemistry is not easy for me. Okay, so two main modes of wood decay, white rot and brown rot. So in white rot, the substrate is white. After decay, in brown rot, it's brown. White rot is sort of the brute force mechanism. The fungi attack the lignin and all the carbohydrate components of the plant cell wall. And these class 2 peroxidases are really important, we think, in lignin decomposition. Brown rot is this really elegant mode of decay where the fungi have figured out a way to circumvent lignin and use a non-enzymatic strategy for pulling out cellulose, for initiating depolymerization of cellulose. And so when the substrate is left over, that's mostly lignin. It's been modified, but left largely intact. This, in contrast, is mostly crystalline cellulose, the residues there. This is the oldest white rot fossils from the Permian about 250 million years ago, or the Triassic about 225 million years. So white rot is fairly old. Basidiomycetes are older. These are hyphae with clamp connections, diagnostic for basidiomycetes from the Pennsylvanian. These are mycorrhizal roots from the Eocene. These are modern things, so about 50 million years old. We have some fruiting bodies. This is the oldest from about 130 million years old. It's a polypore. Might have looked something like this fungus here. And here's a picture from my very brief career as a paleomycologist. This is a Cretaceous mushroom from amber from New Jersey, of all places. So fungi do, and agaricomycetes do really important things, right? And they've been doing them for a very long time. But when we tell the story of the history of life, we typically just ignore the fungi. So this comes from the textbook that we used to use in the intro bio class I teach. Fungi get one little mention here, but other than that, from here on up, it's all, charismatic megafauna and vascular plants, right? So this is a little bit like trying to explain the history of humans strictly in terms of wealthy, powerful white men. It's incomplete. So now I want to talk about coal briefly. Coal is a mega commodity. We still get about 40% of our electricity in the United States from burning coal. About 6 billion tons a year consumed worldwide. And the coal people tell us that we can be mining coal for a very, very long time. And coal, as I'm sure you all know, the vast majority of coal reserves were deposited during an interval about 360 to 290 million years ago in the Carboniferous period, when we had these arborescent seedless vascular plants, so relatives of club mosses, ferns, horsetails, things like that. There were also seed plants in these ecosystems. And these deposits gave rise to coal. So, why? What was so special about the Carboniferous period? So if you want to know the answer to this question, or if you want all the good news about coal, you can go to the website of the World Coal Association, and they give you, which I think is sort of the standard explanation. This is purely explained in terms of abiotic processes. So they say, when plants die, solar energy is usually released. So it kind of implies that decay is a continuous process. It's something that's always there, always happens. But under some conditions favorable to coal formation, this decaying process is interrupted, and all that organic matter ultimately becomes coal. So this is explained strictly in terms of climate, tectonics, abiotic processes. Now, an alternate view was proposed by this woman, Jennifer Robinson, in 1990, the same year as that famous paper, another paper that's had a big impact on me, although only more recently. And what she suggested is that changes in the biotic component of the environment might have also influenced rates of coal deposition. So here's the Permo-Carboniferous peak and then collapse in organic carbon sequestration. Robinson was basing her inference on a very scanty number of basidiomycete fossils that start to crop up in the tertiary. She also made an inference which now is really very questionable, and suggesting that these arborescent lycopods were particularly enriched in lignin, that heterogeneous recalcitrant polymer that we think is a major component of coal. Not the only component, but a major component. That conjecture now we're questioning, but nonetheless, that was part of her hypothesis. So here's the Robinson hypothesis in full. She says, as dominance shifted from lycopsids and pteridophytes, blah, 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 basically there was a shift in plant investment in lignin. So plant architecture became more sparing in its use in lignin, and lignin-degrading organisms were rare or absent in the Paleozoic, but diverse and abundant in the Tertiary. So suggesting that the evolution of white rot fungi may have contributed to this decline in carbon sequestration. Okay? So with that introduction, general questions that I'll address today is, what's the ancestral mode of wood decay in agaricomycetes? What are the genetic correlates? And really the big question, or a big question, is could the evolution of white rot have contributed to this Permo-Carboniferous crash in carbon sequestration? There'll be two parts to the talk. One is going to be based on a somewhat old paper now that some of you have heard me talk about before, and then the second part will be on some newer stuff. So we'll go on to first recapping this paper by Dimi Flutis and colleagues. This was a comparative genomic analysis. We wanted to understand the evolution of white rot and brown rot. There were 31 genomes for the time, that was a large analysis. 22 species and 10 orders of Agaricomycotina, the Agaricomyces, and then a bunch of fungi from outside of that group. And we tried to sample, white and brown rot across as many clades as we could. But from the gray letters, you can see that, there's a lot we didn't get to. We made trees. I'm a systematist. We make trees. Had a bunch of different data sets, a bunch of different analytical methods. This is Dimitri. The tree is quite robust, although we know now that when you have scads of data, you can get basically false positives, 100% bootstrap support for nodes that are wrong. Nonetheless, we're reasonably confident in the tree. You'll see this tree in a bunch of slides. So from here on up, it's the Agaricomycotina. Brown is brown rot, white is white rot, one ectomycorrhizal species, a couple pathogens, or animal and fungal parasites here. Okay? So we made a tree, but what we're really interested in is understanding the evolution of the gene families involved in decay. So we annotated the 27 selected gene families, and this is the result. So here are white rot species, brown rot species, the one ECM, oxidoreductases involved in decay, CASEs, warmer colors or higher counts, and so on. So, and then here are the average number of copies and the average number of families represented. So white rot species have 140 copies on average in 25 families. Brown rot, about 74 copies in 18 families. So brown rot fungi are depauperate in all of these classes of enzymes. And we're going to focus just on a few individual gene families. So the class 2 peroxidases here, which you can see are really strikingly different in their distribution across white rot and non-white rot species. So here's the tree, and the bars are the copy numbers, 14 on average in white rot, 1 or 0 in brown rot, and they're basically absent from all other fungi. I'll talk about these in a moment. These are Ascomycota out here. We're also interested in cellulose because that's the other major component of plant cell walls. So we looked at cellobiohydrolases and then these LPMOs, also formerly called GH61, which we think are important in attack of crystalline cellulose, which is in itself a very resistant, hard structure to decompose. This is a cellobiohydrolase cruising along a strand of crystalline cellulose. It's got a carbohydrate binding module that helps it adhere to the surface of the crystalline cellulose, and it's cleaving off cellobiose as it marches along. This is the trichoderma cellobiohydrolase, the industrially important enzyme. So when we look at GH6, GH7, and then the LPMOs, we see a pattern that's really similar to the class 2 peroxidases, expanded in white rot, contracted in brown rot. We can also look at the CBMs, those cellulose binding modules. We calculated the number of those modules across white rot, brown rot in 8 different CASE families. And whether you look at the total number of CASEs or the percentage of the genes that bear a CBM or enzymes that bear a CBM, it's the same pattern. They're expanded in white rot, contracted in brown rot, but also present in Ascomycota. Okay? So we'll go back to class 2 peroxidases for a second because they're so important. We have these counts on the tips for the species, gene counts for each species. But what we want to know is, we want to reconstruct the history of the evolution deep in the tree. And so for that, we use the gene family phylogeny. So this is the phylogeny of the genes that encode those peroxidases. There are a few class 2 peroxidases in Ascomycota out here. But based on structure function considerations, we think, when I say we, I mean Angel Martinez, my collaborator, who is much more knowledgeable about these enzymes than I am, thinks that these are probably non-ligninolytic. There are also non-ligninolytic pods within Basidiomyces, like the Coprinopsis cinerea pod, for example. The ancestor of the Agaricomycete class 2 peroxidases was probably something like a manganese peroxidase. There are also lignin peroxidases. So we have a pretty good idea about the history of these things, and we think that the ligninolytic pods are limited to Agaricomycetes. But this is a gene phylogeny, right? And it's got a lot of weakly supported nodes, so this still doesn't directly give us counts on the organismal tree, right? It doesn't tell us how many copies the ancestor of the Agaricomycetes had, for example. So to get at that, we use gene tree, species tree reconciliation, which are a family of methods that take a gene tree and try to fold it onto a species tree in a manner that maximizes goodness of fit, to put it in simple terms. And we use 3 different methods. And now the dots, the colored dots on the nodes, are inferred copy numbers of class 2 peroxidases. So gray is 0, white is 1, and so forth and so on. And these 3 different methods, they provide quantitatively different results, but qualitatively similar. So we've got a few copies in the ascomycetes, and if we assume they're homologous with those in agaricomycetes, which we do, and if we discount the possibility of horizontal transmission, which we do, we reconstruct 1 copy in the backbone in the ancestor of the Dicaria, the common ancestor of ascomycetes and basidiomycetes, 1 copy through the backbone of the basidiomycetes, and then diversification beginning around the origin of the agaricomycetes. So the initial expansion in the gene family here, the origin of the ligninolytic pods, the pods that can attack lignin, and then dramatic expansions leading to white rot lineages and crashes leading to the brown rot and also the ectomycorrhizal thing. And it's the same picture with the 3 different methods, although the numbers vary. And that's shown here in this figure. It's the same results that you saw before. Now only the vertical height is copy number, right? And it's the same tree stretched out on its side. So an initial expansion in the agaricomycetes, further expansion toward the white rot lineages, and then crashing down in the brown rot lineages. So expansion, contraction in all of the different methods, although the absolute number of copies varies. These are really hard problems. So what we think now is that the ancestor of the agaricomycetes might have been the first white rot species. And we want to know how old it was. So we used molecular clock methods. For calibration, we used a couple of the fossils you saw before and 1 that you didn't see before, but we've got 3 fossils for calibration, bunch of different analyses. With BEAST, we calibrated with all 3 fossils or the 3 different runs excluding one or the other to try to get a sense of if our age estimates are sensitive to any one of these fossils. And the answer is that we get pretty much the same answer with all methods. Huge error bars, right? So these are the 95% highest posterior density intervals, confidence intervals. They're quite wide. So like with all fossils in BEAST, the average age of this ancestor was 290 million years, but look at the range, right? And so we can't reject an age within this interval. Nonetheless, these age estimates are consistent with the fossil record. So if this is the origin of white rot, it's younger than the definitive white rot fossils, the oldest definitive white rot fossils. And it's younger than the oldest definitive basidiomycete fossils. So at least these fossils, which we didn't use as calibration points, don't reject these results. And they're consistent with the Robinson hypothesis, right? So this is the average age. And of course, the error bars go like from here to here. But that's the average age, and it's roughly coincident with this crash in carbon sequestration. So for this first part, answers to these questions, what's the ancestral mode of decay in agaricomycetes? We think probably white rot, but with some caveats because of sampling, which I'll get to later, but probably white rot. Genetic correlates of the white rot, brown rot modes, expansions of class 2 peroxidases, and other decay-related oxidoreductases and cases in white rot, and crashes in all of those gene families in brown rot, and also in mycorrhizal things. And so could the evolution of white rot in agaricomycetes have contributed to this decline in carbon sequestration? I think this is plausible. And so in the end of our paper, we wrote this rather tortured sentence or paragraph. We were trying to be very careful. And we noted that carbon sequestration crashed at the end of the Permo-Carboniferous, and that frequently abiotic factors are cited as the cause of this. But we said our results are consistent with a complementary hypothesis, complementary, right? Which posits that the sharp decline, blah, 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 was caused at least in part by the evolution of lignin decay capabilities. We're not addressing changes in plant chemistry in this study. We're not addressing history of plate tectonics or climate models or anything like that. And the error bars on those node ages are huge. And gene tree species tree reconciliation is dicey. So we were trying to be cautious, which I think we should be as scientists, right? So this got out into the blogosphere, and which doesn't happen to me very often. This interviewer actually spoke to me, very nice article in Scientific American, and the headline was "White Rot Fungi Slowed Coal Formation." I would have preferred if he had used a title like "The Evolution of White Rot Fungi Could Have Contributed to the Decline," but anyway, but it's the reporter you don't speak to or the blogger you don't speak to that gets you. And so we got why there's no new coal reserves when why there's no new coal when reserves run out and how that could help biofuels. Coal-killing fungus could boost biofuel production. And then my all-time favorite from the Deccan Herald was mushroom culprit for coal shortage. Okay, so we didn't say any of this. Is a typical blame the fungus story, right? So we didn't say any of these things. And I'm showing you these because they're funny and you laughed, thank you. But also to make a serious point, which is that scientists really need to be ready to engage with the press, right? It's important because they'll get it wrong. But here's a headline or title that I take much more seriously. This is a paper that just came out this year in PNAS, and the title is "Delayed Fungal Evolution Did Not Cause the Paleozoic Peak in Coal Production," from Kevin Boyce's group at Stanford. And they say the Carboniferous Permian peak and subsequent decline in coal production likely reflects a unique combination of tectonics and climate. And I wouldn't say that those factors weren't important. But then with the particular details of the evolution and plant and fungal community composition bearing no direct relevance. So saying that the biotic factors had nothing to do with this. And they actually have some really good arguments. This is a superb paper, which I urge you to read, and all 138 citations therein. So the arguments why fungi, the evolution of white rot, could not have contributed to this event, the decline in carbon sequestration, there are several parts of this. One is that lignin decomposition could have occurred before the Permian. They note that these error bars are huge, right? Absolutely, right? We can't reject that the white rot fungi are much older or much younger than the average. This is totally valid. Our best estimate is that they evolved around here, but we can't reject earlier or later origins. Also, they say, this is a quote from the paper, although the earliest definitive fossil record of white rot is in the Triassic, an earlier evolution of lignin decay can't be rejected because we have fossils from the Devonian that show what look like lignin degradation in plant cell walls, okay? So this looks like it could be lignin degradation, even though it's not clearly white rot. Also, there are other fungi besides white rot agaricomycetes that may degrade lignin. So some of the fungi we didn't sample, some early diverging agaricomycetes, certain dacromycetes, ascomycetes, bacteria could decay lignin to some extent. This is a dacromycete fungus, a thing called dacromycete, or Colosera rather. A paper in 1983 suggested that members of this genus might have some capacity to decay lignin. It's the sister group of the agaricomycetes, and that would push lignin decomposition back. And then furthermore, lignin decomposition must have occurred before the Permian because, thinking of simple mass balance, if there was no lignin decomposition at all, then that would result in the complete removal of atmospheric carbon dioxide in less than 1 million years. So there has to have been some degradation of lignin. It's just impossible for there not to have been. And then furthermore, coal deposition continued after the Permian, right? So this is the Permian, and here is, from their paper, organic sediment deposition over geologic time. So, right, so coal deposition continued afterwards. So my response to this is that nobody is suggesting that there was no lignin degradation prior to the origin of white rot, because other fungi can do it, although we think that white rot agaricomycetes, at least in contemporary ecosystems, are the major players. And also nobody is suggesting that coal formation ceased, that organic carbon sequestration ceased after the Carboniferous. So it's a rather simplistic model. I think the question really is about the relative rate of productivity versus decomposition and carbon sequestration, which is a different, somewhat more nuanced question. The Nelson et al. Paper goes on and says that lycopsid paraderm was probably not lignified. So even if lignin is tremendously important, based on some very high-tech X-ray synchrotron methods that I don't fully follow, the paraderm of lycopods, these trunks, these were weird plants. They really didn't have that much true wood. Most of the mass of the plant was what we would consider like bark-like material. And based on these analyses that have been published by others, that may have been heavily enriched in suberin, which is a more aliphatic, less aromatic organic material, right? Which in fact some fungi can decay, right, with carbohydrate esterases. Nonetheless, if this really was the major component of a major component of coal deposits, then lignin decomposition might not be expected to have a huge impact on carbon sequestration. And then finally, there's no clear impact of distinctly different biochemical signatures that successive swamp assemblages would have generated. So this is a figure from a paper by, I think it's pronounced as DeMichael and Phillips, showing that the composition of coals has changed over time. It hasn't always been dominated by lycophytes. At times it's been dominated by ferns. Some gymnosperms, the chordaides, may have been important. And these, for example, would have been much more heavily lignified probably than the lycopsids. And if you assume that lignin is the primary determinant of coal deposition, you would expect a spike in coal formation as the chordaides become more prominent, and you don't see that. So in response to this class of criticisms, I would say that I can't explain the vagaries in coal deposition. I think it's probably very complicated, but I would like to point out that white rot is not just about lignin. And I think I'm guilty of overemphasizing lignin in that science paper because it's a telegram and you only get a few words. But yeah, we did overemphasize lignin, but white rot is not just about lignin. So recall that these enzymes acting on crystalline cellulose also appear to be expanded in white rot. If you dig into the supplement of the paper, we did reconstruct the evolution of lots of different families. So we had about 11 or 12 families that we think expanded around the origin of the agaricomycetes, which is here. I'll zoom in on this. And they include, in addition to class 2 peroxidases, didecolorizing peroxidases, heme thiolate peroxidases, enzymes that generate peroxide, various cases, pectinases, the LPMOs, lactase, et cetera. So it looks like a general elaboration of the decay apparatus, perhaps triggered by the evolution of pods, maybe, but not just the evolution of lignin decay. But this is still based on a handful of gene families and really a small number of genomes. And so this brings me to the last part, where we'll talk about some of the more recent studies led by a former postdoc, Laszlo Nagy. One paper published this year in MBE, and another that is struggling through multiple submissions to various journals. So this is the tree of Agaricomycetina on microcosm, the remarkable JGI genome portal. So the red arrows here are the genomes that we sampled in the Flutus et al. Study. So we tried to spread, I think we did a pretty decent job sampling diversity, but not so much in the early diverging groups in particular. So in the Nagy et al. Studies, we now have 62 genomes, 51 in the Agaricomycetina, adding these blue arrows in addition to the red arrows representing the Flutus genomes. And in particular, we did a lot of sampling in the early diverging Agaricomycetina. And so this lets us sort of focus in on the evolution of this complicated white rot apparatus. And so we can show that these two new colosseras that we got in here, one of which had previously been reported to be white rot, looks like a typical brown rot fungus. So based on genomic comparisons, we see no evidence of lignin degrading capacity in these fungi and in other Cantharellales and Sebaceales. The expanded genome sampling also lets us get a more precise picture of the evolution of genes involved in lignocellulose decomposition. So here are the pods. This is the origin of the, this is the origin of the Agaricomycetes down here. Previously, we had not sampled these lineages. And so we thought we were reconstructing this node here. So we see the expansion of the pods, which is the darker colors or higher copy numbers, around the origin of the agaricomycetes. But the LPMOs, these lytic polysaccharide monooxygenases that we think are important in attacking crystalline cellulose, they're older, right? They're in ascomycetes, so they go way back. But they also expanded in agaricomycetina, but a little earlier. So it looks like they expanded earlier. And we see a similar pattern if we sum across a bunch of bulk cases that attack major plant cell wall carbohydrates, right? So they're old, they're in the ascomycetes, but they also expand early in the origin of the agaricomycetes. And then the last tree like this I'll show you is a group of enzymes I haven't spoken about. These are the P450s, cytochrome P450s, which are very diverse, not that well understood, but thought to play a role in detoxification of xenobiotic compounds, maybe lignin degradation compounds, and so on. Hundreds of copies in agaricomycete genomes and in others. And again, this is a group that we think expanded around the origin of the agaricomycetes, right? So a general elaboration of this decay apparatus, and not all happening all at once. So this is all fine, but this is still, the known, not to get Rumsfeldian on you, but this is sort of talking about the known knowns, right? So we've been talking about gene families that we know or suspect to be involved in lignocellulose decomposition. What we'd really like to be able to do is really harness the power of the genomes and the phylogeny to try to get a sense of the total package of gene families that might be important in the evolution of white rot. And that's in this paper that we're currently struggling with. And so it uses the same genomes as the prior study. And it uses a bioinformatics pipeline that Laszlo Nagy, this former postdoc, developed, which is called COMPARE. And the basic idea with COMPARE is that you're trying to identify gene family expansions and contractions that are correlated with the gain or loss of a function. So gain would be blue, red would be loss. And so if lambda and mu are gain rate and versus loss rate, you can predict that if there's a gene family that has a function in a particular trait, and if gene dosage or diversification is important in that trait, that you should expect to see enhanced duplication rates when the trait is on, and reduced when the trait is off. So the pipeline that Laszlo created, basically the outline is like this. So first you do ortholog coding, so you take all of the genes in the genomes, divide them into gene families, and then divide them into orthogroups, which are groups of putative of orthologous genes. Map those onto the organismal phylogeny with Dalo parsimony, and then map the trait onto the organismal phylogeny, estimate the diversification rates that are specific to the presence or absence of the trait of interest, and then there's an ANOVA to assess significance. And this is kind of a cartoon of the whole process. So we did this, and these are kind of the results. There are about 1,600 families that look like they might be correlated with white rot. When we look at the families that are then secondarily lost in the non-white rot lineages, so brown rot and ECM, we're down to about 408 families. 310 of those have homologs in Phanerochaetae, which is sort of the model system for white rot decay in basidiomycetes. And of those 310, 190 or 62% have been shown to be upregulated or have at least one member upregulated in experiments when wood is the sole carbon source as opposed to glucose. So we have some transcriptomic corroboration that we may at least be on the right track for some significant fraction of these gene families. So what are they? It's an omics study, so you get these horrible long tables. And I'll summarize what they are. So what we pull out now is the expected peroxidases, so PODs, dipes, hemethylate peroxidases, lactases, diverse cases, P450s, and glutathione S transferases, and then transporters, right? And 73 gene families with no known PFAM domains, of which 26 have at least one member upregulated in Phanerochaetae. So we think this may be an approximation of the white rot toolkit, to use that expression. Okay, so that's all the evidence. So to conclude then, what about the evolution of plant cell wall decay in agaricomycetes? Well, the ability to degrade cellulose and other carbohydrates, these very ancient components of plant cell walls, is itself very ancient, present in the ancestor of the Dicaria, the ascomycetes, and the basidiomycetes. Efficient decay of lignin is a much more recent innovation, with these ligninolytic pods arising in the most recent common ancestor of a group called the auriculariales and all other more derived agaricomycetes. There was this general elaboration of this plant cell wall decay apparatus early in the evolution of the agaricomycetes, and not all at the same time. White rot is conserved in the backbone of the agaricomycetes, but then with parallel origins of brown rot and ECM, there have been repeated losses across many of these gene families, so gains and losses, as agaricomycetes sort of shifted away from this brute force method of white rot toward the more sophisticated brown rot and ectomycorrhizal nutritional modes. Well, what about white rot and coal deposition? Well, I think that this simple model in which there's no lignin decay before the evolution of white rot and there's no coal formation afterwards is just not plausible. So I think that the Nelson et al. Paper very strongly showed that this simple hypothesis is just not supported. I think the strongest evidence for that is that the inference that this lycopsid paraderm maybe didn't really contain very much lignin, and the mass balance analyses suggesting that the absence of lignin decomposition at all is just impossible. It would have pulled all the CO2 out of the atmosphere. But I would say that the rejection of that simple model does not preclude the idea that the evolution of this complex white rot mechanism could have contributed to this decline in organic carbon sequestration, in part because white rot, it's not just about lignin, it's not just about class 2 peroxidases. So to really hammer this question, I think we need to rethink the questions. So, important outstanding questions are, when did white rot evolve? We need better estimates of the age of this node. We need more fossils, better molecular clock methods. We also need to think about other ways to reconstruct gene family evolution. This gene tree, species tree reconciliation is really tough. We also need to ask, what would the impact of white rot have on organic carbon sequestration? And I think this requires really sort of a quantitative modeling approach that takes into account climate, tectonics, and the biotic components of paleo ecosystems, including the decay capacities of fungi and the biochemical components of plant cell walls. So easy for me to say, but not easy to do. So currently, I still think it's plausible that the evolution of white rot contributed to this decline in coal formation. And if you have a hard time envisioning that, think about, these are white rot fungi. Think about what they do in contemporary ecosystems. These are fungi that are able to take a solid block of wood, in this case this is an oak, and reduce it to a stringy mass of cellulosic pulp that other fungi and bacteria can then degrade. So think about the impact that it would have if you removed these organisms from contemporary ecosystems or added them to an ecosystem like this one. Now, it's possible that the impact would have been very modest, right? Or it could have had a large impact. And I'm not necessarily the paleoecologist or carbon cycle modeler to be able to resolve that. But first principles suggest to me that the evolution of white rot in these systems should have had an impact on carbon sequestration. I may not have convinced you of that, but I hope I have convinced you that we cannot understand the functioning of contemporary ecosystems or their evolution unless we consider fungi. So thank you.
So we have time for questions, but I'm reminded that I should mention that we have a reception after this in 338 that you're all welcome to. And there's also a film of this that you can review later. So with that, questions for David?
So I've not read this other paper about the mass balance, but did they take into account things like fires and volcanism that turns limestone back into CO2?
Yes, yes. They specifically discuss weathering. And I think actually it's a very strong argument that, that if you had no lignin degradation at all, if it was just absolutely all sink, that would have had catastrophic impacts. But my reading, and this is very dangerous for me because this is not my field, right? But my reading of that stuff also suggests that small shifts in rates of lignin decomposition could have had large impacts on carbon sequestration. Yeah.
So there's three common ancestors that are often seen as saprotrophic: the Dicarian common ancestor, the Basidiomycete, and the Garicomycete. I know you're focusing on maybe the Garicomycete, but is there any, do you have any insights into the different type of saprotrophy that those three had, or how long?
Yeah. Well, first of all, I don't know if they were necessarily all saprotrophic. They could have been plant parasites as well. But the attack of cellulose and other carbohydrates is very old. So this mode of decay in which you have direct attack on crystalline cellulose with cellobiohydrolases and so on seems to be plesiomorphic, right? But then within the agaricomycetes, then you had the evolution of the class 2 peroxidases and other enzymes that enabled that to be coupled with attack on lignin. But then subsequently, you have a couple of other things that happen: loss of the class 2 peroxidases and reduction in the cellobiohydrolases, et cetera, in brown rot as they evolved this Fenton chemistry to pull out cellulose. But some agaricomycetes have lost the pods and so on, but retained the cellobiohydrolases and seem to have gone back to what I think might be a plesiomorphic kind of decay, emphasizing direct attack on cellulose, but not a very efficient lignin decay mechanism. This is all conjecture on my part, really. Yeah.
So in your analysis of the gene family evolution, you've talked mainly about gene family expansion or loss. And in those expansions, do you think those expansions are mainly due to just having more is better, or do you also see signatures of diversification?
Yeah, that's a great question. So the question is is it just gene dosage or is it neofunctionalization? And I really don't know, because I don't think we know enough about the differences in the functional attributes of those different copies. So short answer is I don't know, but longer answer is that diversification does seem to be correlated with white rot, so increasing copy number. And we can test that using some methods that look at other methods besides the ones that Lazo developed, looking at diversification rates correlated with different traits. So speciation rate of class 2 peroxidases is elevated in white rot lineages relative to brown rot. But whether that's because of new functions evolving or just gene dosage, I don't know.
So the Dacromycetales seem like a weird set, right? Because they're primitively brown rot,
Right.
And in all other groups, the brown rot is derived.
Yeah.
So any insight into their origin, sort of how they and why, if brown rot's an advantage, why didn't they diversify more or something, or is it?
Yeah, I have no idea. I mean, we think that that's a lineage that split off before these ligninolytic pods came into existence, but how they convergently evolved this Fenton chemistry, I don't know. I don't actually know how much is really known about the chemistry of Dacromycetalian brown rot. I mean, brown rot has evolved repeatedly, so there's no reason to assume it's the same in all groups, but you can understand why it might have evolved repeatedly, because it lets the fungus avoid the energetically expensive work of breaking down lignin to get it at carbohydrates. That's all very sort of hand-wavy. And why there aren't more Dacromycetes, I have no idea. Maybe because they never evolved gilled mushrooms or something. Maybe it has nothing to do with decay. I have no idea. Yeah?
Have there been any kind of published, hypotheses as to like the rate of buildup of the organic matter of the plant material to sort of fine-tune when it's sped up and slowed down?
Yes, I mean, yes, and it's from a couple different directions. There's like sedimentology that's been done, and that's way away from anything I do, so I'd refer you to the Nelson paper. There's also been there have been carbon cycle models that have been developed, several different families of carbon cycle evolution models that explicitly model things like the rate of carbon sequestration, which is not which I think does get to your question. So yes is the short answer from both just actually examination of the sediment record and also modeling studies.
The reason why I said is you have to have the plant material before you have the things that break it down, right?
Sure. Yeah.
Is coal formation always associated with an origin in that group of plants, or are there any
No.
lignin-containing plants that also lead to coal formation?
I don't think there are any non-lignin containing plants, but I think what Kevin Boyce is arguing is that these lycopsids, the arborescent lycophytes, had much lower lignin content than, for example, Jennifer Robinson thought. So the paraderm, he suggests, is heavily enriched in suberin, but not lignin. They had a relatively small strand of secondary xylem, which would have been lignified, of course. I don't know if there are any plant groups that do not contain lignin that are thought to be major contributors to coal. But the relative amount of lignin, I think, varies a lot. And so one of the arguments is that if lignin is so important, then you should see coal deposition rates waxing and waning as the plant assemblages change. Hard to tease out, I think. Yeah, Iman, did you have a?
Yeah, I was interested in your relationship with the press now. You said like it's really important for scientists to talk to the media, and kind of what is your strategy?
It's not like they come around all the time. But I'm always happy to talk to reporters on those occasions when they do show up, because it is important that we scientists communicate what we do in a manner that is not only intelligible but makes the work seem compelling, because that helps our legislators on one side of the aisle and our science agency administrators to argue for the funding that we need. So yeah, you got to do it. It's really important. Yeah.
So is do we know, I know you mentioned that lignin is a diverse molecule. Has that always been the case? Is there a chance that maybe there was one type of lignin and then
Yeah.
figured out how to degrade that, and then as a result of that, then there was a diversification of types of lignin, and then a subsequent diversification of...
There are different kinds of lignins. They differ in the subunits, the different kind of these phenylpropanoid subunits, the relative ratios of the different kinds. But I have no idea about fungal adaptation to different kinds of lignin.
I think if there's other questions, you can catch them at the reception, and I invite you all to attend it.
Okay, thank you.